starting to get a lot of tests in classes. It's that window when, professors have covered enough material that it's time to test you on it. So my apologies that you're probably getting more of those this coming week. You're getting them this week or next week for the most part?
I've got like projects due.
Projects due?
Today.
Okay, all right. So just a reminder, you have a test in here on Monday. It'll be over whatever we cover through the end of the day today. There's going to be a homework assignment for you to work on. It gives you some practice doing genetics problems. So what we're going to be doing today is we're going to be talking about genetics. We're not going to get through, why did I bring my phone to class? We're not going to get through all of the genetics that we usually get through before we get to the first test, because we're running a little bit behind. So when you look at exam 1, so there's on the Moodle site, there is an old exam 1, I think it's from 2017. It's from 2019, might be 2019. Anyway, there's one on there. If you are going through that to kind of get an idea of how I go about testing you, which you should do, I put it there for a reason. 2 things it does for you is it allows you to kind of take a practice test if you want to do that. It also will give you some insight into how I go about asking questions and things like that. So it would be beneficial to you to go and look at that. But there may be some questions about genetics that don't make any sense to you because we didn't get that far in genetics. And in that case, you can just ignore those. We're probably going to get through the genetics of single traits today. And I do not know why this is not working. Where is the manual power button on it? So take a look at that, but if you have better. If you have a section on there and you're like, I have really no idea what he's talking about on this question because you haven't seen that in class, chances are it's something that we just haven't gotten to yet, and we'll get to Wednesday of next week. So that said, do you have any questions with kind of what we've been doing so far? Our goal with this is that ultimately you need to understand this process by which we get from genes to this thing that biologists refer to as the phenotype. The phenotype is just the end result of the action of a gene. So for example, you might have blue eyes. Well, the color of your eyes is due to proteins that are present in your irises. The pigments are proteins, and those proteins are determined by genes ultimately. So this process by which genes get converted into proteins is an important aspect of the biology of an organism, because that's how we get eye color, that's how we get hair color, height, it's how we get drought resistance in corn, it's how we get protein in rice, et cetera, et cetera. These are the things that happen. So this process of going from genes to the outward expression of the genes, which is sometimes how we refer to the phenotype, is important to then also understand how genes get from one generation to the next. So we studied mitosis mainly because it gives us insight into meiosis. Meiosis is the way in which we get genes from one generation to the next generation. What we were talking about at the end of class last time is how we then get from a gene to a protein. And so all these things are connected in order for us to understand, for example, how do we get drought resistance and pest resistance and pesticide resistance, for example, into a plant cell that wasn't there to begin with, so we can start talking about genetic modification. In order to understand genetic modification, we have to understand all these things about genes. So we were going through this thing that is called the Central Dogma. It's not necessarily a term that I like, but it's kind of the term for it. But it's the process by which we go from the genetic information in DNA to the genetic information in messenger RNA, to the structure of a protein. And it's basically this is moving from genes in your DNA to proteins, which is essentially what influences your phenotype. So, we talked about transcription. This process is called transcription. Transcribing DNA into RNA, and then this process is referred to as translation. And I don't think I gave you these terms last time, but they're useful because they're essentially an analogy. They're the terms that we use in biology to describe these processes, but they're also useful as an analogy. When you transcribe a book, what are you doing? Did you guys ever see the movie The Name of the Rose? Oh my gosh, The Name of the Rose? Sean Connery plays this Franciscan monk. He has an acolyte played by Christian Slater, a very young Christian Slater. And it's a murder mystery. It's from a novel written by a guy named Umberto Eco, an Italian author. It's set in the Italian Alps, and it's set at this monastery where there's a scriptorium. This monastery is famous for having this amazing library. And all of a sudden, the monks are dying in mysterious ways, and the character played by Sean Connery is I think it's Sir William Baskerville, and he's kind of a Sherlock Holmesian type of detective priest person. And in the story, all of these monks in this monastery are scribes, and they sit at a desk day after day for hours at a time, and they basically have a manuscript, a book, and they have a book that's already written, and they have a book that is filled with a bunch of blank pages. And they're basically copying this book into this book, and this is how we reproduced books before we had a printing press. A monk in a monastery would sit and he would copy word for word, illustration for illustration, what was in this book into this blank book, and then after he was done, you would have a copy of that book. And they, of course, made changes that were on purpose. They also made errors that were inadvertent as they did that transcription. So when we talk about transcription, we're talking about the work of scribes taking a book and copying it word for word into a new bound volume. So transcription is moving from a book that's in one language and copying it into a book of the same language. And so transcription is taking information that is stored in the form of nitrogenous base sequences and it's converting it into a new form, but not very new. But the language is still the same. It's the language of nitrogenous base sequences. This is the information content is stored in nitrogenous base sequences. So in this way, you're just transcribing from one DNA code to an RNA code, but it's all nucleic acids. It's all based on this sequence of nitrogenous bases in these linear molecules. But when we move from messenger RNA, and we use messenger RNA as the template for determining the sequence of amino acids, we're moving kind of from one language to another language. And in this case, the language is the language of amino acid sequences in a protein. And so that's not analogous to transcription, it's analogous to translation, taking a book and copying it from one language into another language. Translating a book written in Italian or Latin and translating it into English or Japanese or something like that. And so we use these terms transcription and translation to get the idea that this is a movement from the information content of a nucleic acid to the information content of a nucleic acid. This represents moving from information content in nucleic acids to information content in amino acids. So that's the reason for those terms. And so DNA, the sequence of DNA, the sequence of nitrogenous bases in DNA, are ultimately what genes are. A gene I haven't really given you a definition yet, but I will. This is kind of a formal biological definition of what a gene is. A sequence of nucleotides. Who can remind me what a nucleotide is? Valuable test review. What's a nucleotide? You have the sugar phosphate backbone. To each of these sugars is joined a nitrogenous base. These are then connected to other nitrogenous bases, joined sugars. Joined to nitrogenous bases, add the sugar. What part of this is that I've drawn up here is a nucleotide?
Is it like the center part?
It's not the center part. Remember, this is a polymer. It's made up of bunches of repeating units all strung together in long chains. So the long chain is the single strands that are bonded together. What's the repeating unit within the single strand? Yeah, so it's the phosphorus, the phosphate, the sugar, and the nitrogenous base. This is a nucleotide. This is bonded to another nucleotide, bonded to another nucleotide. Okay? So a sequence of nucleotides that code for a functional product, it's a sequence of these things, and the sequence that is important is what the actual identity of the nitrogenous bases are. Is it adenine, thymine, guanine, or is it thymine, adenine, cytosine, etc.? Those are two different sequences that do different things, code for different proteins. And when we say it codes for a functional product, we used to, back when I was in high school, talk about a gene in simpler terms, and that a gene was a sequence of nucleotides that coded for a particular protein. But we know now that genes code for other things other than proteins. For example, they make messenger RNA and transfer RNA. So rather than make a long list of all of the things that genes are responsible for coding, we just say it codes for functional products. Well, proteins have functions, transfer RNA has function, messenger RNA has function, ribosomal RNA has function. There are all of these gene products that we know about now that we didn't know about 30 years ago. 30 years ago? Oh geez, it's closer to 40 years ago now when I was in high school. So all these different products are determined by genes, and so we have a much more general definition for what a gene is now. So these sequences of nitrogenous bases in DNA get used to specify the sequence of nitrogenous bases in messenger RNA. This long single-strand molecule that is formed in the nucleus of the cell and then gets exported out of the nucleus of the cell into the cytoplasm of the cell. Once it's in the cytoplasm of the cell, it comes into contact with the ribosome. The two parts of the ribosome clamp down on that messenger RNA molecule. Another type of RNA called transfer RNA is the carrier of the amino acids. And it has an anticodon that matches up with the codon on messenger RNA. So we have adenine, uracil, guanine here. It matches up with the uracil, adenine, cytosine on transfer RNA. And that transfer RNA only binds to a methionine.
molecule, a methionine amino acid.
It fits into this active site, this binding site, and there's another binding site right next to it where this transfer RNA is going to come in and join it. This transfer RNA can come in because it has an anticodon that is guanine, adenine, guanine, and that's guanine, adenine, cytosine rather, and it matches up through complementary base pairing with cytosine, uracil, and guanine. It's going to come in here and sit right next to it. Once it's done that, the ribosome acts as an enzyme. It facilitates a bond forming between methionine and leucine. Leucine is this amino acid here. Leucine is the only amino acid that would be attached to a transfer RNA with this anticodon. Once that bond with leucine to methionine is formed, this transfer RNA gets kicked out, it goes back into the cytoplasm and picks up another methionine molecule that's floating around in the cytoplasm. And then this ribosome moves down a slot. It moves down the messenger RNA molecule in this direction so that it's no longer in the second position, it's now in the first position. Now that it's in the first position, the second active site is open so that another transfer RNA molecule can come in here. Once it's in here, this ribosome acts like an enzyme, facilitates leucine binding to whatever this amino acid is. Once that bond is formed, the bond of leucine to the transfer RNA molecule is broken, kicks that transfer RNA molecule, and then it moves down the RNA molecule another spot. That new transfer RNA is now in the first position, ready for another transfer RNA to come into the second position. And it just keeps doing this over and over and over again until it gets to a codon that matches up with a transfer RNA molecule that doesn't have an amino acid attached to it. And at that point, that codon is a stop codon. So we have a table. You do not have to memorize this table. I don't even ask biology majors to memorize this table. But there's a table that basically shows you, remember that codons are 3 sequences of nitrogenous bases in messenger RNA. So you can have, the first base can be uracil, the second base can be uracil, and the third base can be uracil, in which case you have uracil, uracil, and uracil. This codon codes for the amino acid phenylalanine. AUG codes for the amino acid methionine. You'll notice in this graphic, the first amino acid in this protein chain is a methionine, because this codon, adenine, uracil, guanine, is the start codon. It's the codon that your cellular machinery recognizes as the sequence of 3 amino acids, or 3 nitrogenous bases in messenger RNA that tells the cell, I need to start making protein at this point. And that also corresponds to the amino acid methionine. You'll notice that that's the only start codon. There are lots of different combinations. I think there are 64. 4 times 4 times 4, yeah. There's 64 of these. And they code for different amino acids. There's one start codon, and you'll notice that there are 3 stop codons. Uracil, adenine, adenine, uracil, adenine, guanine, and uracil, guanine, adenine. Those stop codons complementary base pair to transfer RNA molecules that just differ from other transfer RNA molecules in that they don't have an amino acid attached to them. So when they get to this point in the protein synthesis process, rather than there being an amino acid out here, there is no amino acid. The ribosome wants to add this leucine to another amino acid, but there is no amino acid there. And so what eventually happens is the ribosome just lets go of the messenger RNA molecule and then kicks that amino acid sequence out. But in the end, what you get is you get this long chain of amino acids, which is the basic structure of what a protein is. It's just a long chain of amino acids. there are 3 stop codons. But you'll also note, what else do you notice about the things in this table? How many ways is proline specified for? Proline is right here. Do you see proline anywhere else? I only see it here. Specified in 4 different ways. But it's specified by cytosine, cytosine, and anything in the 3rd position. Leucine. Leucine is specified 6 different ways. In one case, leucine is specified by cytosine, uracil, and anything in the 3rd position, and uracil, uracil, and either an adenine or a guanine in the 3rd position. There's a lot of redundancy in the genetic code, and a lot of the redundancy is such that it doesn't really matter what's in the 3rd position. All that really matters is what's in the first 2 positions. And that's enough to specify a particular amino acid. This could be cytosine, cytosine, uracil gives you proline. Cytosine, cytosine, cytosine gives you proline. Cytosine, cytosine, adenine gives you proline. Cytosine, cytosine, guanine gives you proline. We have 64 different codon sequences to specify about 21 different amino acids. And so there's this redundancy in the code. One of the nice things about redundancy in the code is it prevents your cells from making mistakes. And as it turns out, the 3rd spot in the codon sequence is also the most variable spot, because over time evolution has not selected against organisms that have mistakes in that 3rd position. So you find that there's a lot of variability in the 3rd position of this codon structure. When we talk about things like mutations, a lot of mutations occur in this 3rd position because it doesn't matter what's there. And so those mutations stick around. So protein synthesis starts with a start codon. So the first amino acid in your protein will be methionine, and then it ends when it encounters one of these stop codons. And then what's between the beginning and the end is all determined by what the sequence of nitrogenous bases is in the messenger RNA molecule, in this long messenger RNA molecule. Questions about that process? I've talked to you about processes in the past. The easiest way to learn about processes is to learn them as a process. So learn this in a series of steps. What's the first step? Well, you have DNA. It opens up with an enzyme. That opening up exposes the nitrogenous bases of DNA, and then those nitrogenous bases allow messenger RNA nucleotides to come in. The messenger RNA nucleotides bond to one another. And that RNA strand separates from the DNA. RNA, the messenger RNA, goes from the nucleus to the cytoplasm. Then the ribosome captures the messenger RNA, and at that point, protein synthesis begins. At the start codon. Et cetera, et cetera. It's a series of processes. You can't have this happening unless you've gotten messenger RNA formed. You can't have messenger RNA formed unless you've opened up the structure of DNA so that the messenger RNA nucleotides can be built on the template that is set forth by the sequence of nitrogenous bases in DNA. So learning it in order, in a process, is really the best way, the only way to learn it. And so, once again, if you need to rely on pictures to learn it, if you need to write out the process in words to learn it, learning it as a process, whichever one of those ways you need to do is the way to go about it. Biology students oftentimes, when they get to things like this, they want to just memorize all the names of things and somehow think that in the process of memorizing those names and knowing what messenger RNA is, et cetera, that will get them to learning what the process is. But they struggle with that because they're not starting at the beginning with the focus being learning the process. The process isn't finished because there are lots of details about protein synthesis. We have transfer RNA coming in, codon matches up with anticodon. Where's the anticodon? Is that on messenger RNA or transfer RNA? Are anticodons on transfer RNA or messenger RNA? The answer is on the screen.
What was the question?
Is an anticodon on transfer RNA or messenger RNA?
mRNA.
This is transfer RNA, this is messenger RNA. What's on the transfer RNA?
Oh, I was looking at the codon.
Yeah, this is the codon. This is the three nitrogenous base sequence. In messenger RNA, anticodon is the complement of that in transfer RNA. So this is transfer RNA has the anticodon, which matches up with the codon of messenger RNA. And this transfer RNA has a specific amino acid attached to it. Once the transfer RNAs are there, the peptide bond, which is just the name for a bond between two amino acids, formed. And after that is formed, the first transfer RNA molecule is ejected, and the ribosome shifts. Down the messenger RNA molecule. And then the process repeats until you get to a stop codon, and then the ribosome dissociates, frees up the protein, frees up the messenger RNA. So it's a linear process. At the beginning of class, I was talking to you about how being literate about science is useful, especially in the year 2021. And when I was talking to you about that, I told you about the vaccines that are out there against COVID. And people were worried because these vaccines were messenger RNA vaccines. And people were scared because you're actually putting genetic material into your body from a virus. Which is weird to me because traditional vaccines, they actually put the whole virus in you, in reality. So messenger RNA vaccines seem safer to me because you're not actually getting a whole viral particle, you're just getting a little chunk of genetic material. But the chunk of genetic material that you're getting is messenger RNA. So what happens when a cell needs a particular protein? What the cell does is it activates the gene. The gene, when it gets activated, what it's being activated to do is being activated to make messenger RNA. And it'll make a bunch of copies of messenger RNA. And it ships those copies out into the cytoplasm, and then that enables the cell to make whatever protein it needs to make. Let's say that you just ate a big meal, and you need more insulin to absorb the sugar that is now in your bloodstream as a result of having eaten that meal. The cells of your pancreas will activate the gene that is responsible for creating insulin, which is an enzyme that facilitates the movement of sugar across cell membranes. And your pancreas will start creating copies of the messenger RNA that makes insulin. That will be responsible for specifying insulin being made in the cytoplasm of those cells. Now, as it creates insulin, your body absorbs that sugar, and there comes a point where you don't need insulin anymore. You don't need this pulse of insulin anymore. And at that point, the genes in your pancreas will stop being activated to produce messenger RNA, and then as a result of that, will you continue to produce insulin, or will it stop? It's important if you're diabetic. When you're diabetic, you lose the ability to produce insulin in some cases. You have all these messenger RNA molecules floating around in your cell. Why does the cell just not keep making more insulin after you don't need it anymore? What did I tell you about messenger RNA molecules back there at the beginning of the semester? You might not remember, but see if you can dredge it up. Is messenger RNA a stable molecule or an unstable molecule? Are you guys awake today? Talk to me. Do you not remember? Messenger RNA is an unstable molecule. It sticks around for a short period of time and it breaks down very quickly. If you've been following the COVID vaccine thing, what's one of the difficulties with transporting COVID vaccines? You might have heard about this on the news.
Like temperature control?
They have to keep them really, really cold. Why do they have to keep them really, really cold?
I assume because they're unstable.
If you warm them up a little bit, they start to break apart. I mentioned way back when, I was talking about this earlier, if you're doing a gene expression study in biology, you have to be extremely careful about how you handle samples because there are enzymes all over the environment that break down messenger RNA, and that's what you're trying to actually isolate if you're doing a gene expression study. And so you have to use special pipettes that are only used for messenger RNA extraction. You buy special tubes from the biological supply companies that have basically had all of the enzymes deactivated from them. They've gone through a special cleaning and processing procedure so that there aren't any of the enzymes that are capable of breaking down messenger RNA. In a lab like over in the biology department at Jewel, if you're trying to extract messenger RNA, it's really hard because it breaks down so quickly. Imagine that you have pancreas cells and you need insulin produced. You produce messenger RNA, but then you get to the point where you don't need insulin anymore and these messenger RNA molecules stick around for forever. Then your cell just keeps producing insulin for a long period of time and basically kills you because now you have too much insulin being produced. But one of the reasons that this doesn't happen is that the messenger RNA molecules that it produces that ultimately create insulin through the process of protein synthesis, those molecules are transient. They get formed, they get used, and then they break down. And then if the cell needs more of that protein, it will reactivate the gene that creates the messenger RNA that is responsible for creating the protein, and it'll just make more copies of that, and then it'll get used, and then it'll break down. And if the cell continues to need insulin, it will activate DNA to make the messenger RNA to do that. But as soon as you don't need insulin anymore, the gene, the DNA gene, stops getting activated. It doesn't make the messenger RNA, and it doesn't make more protein. And because the messenger RNA breaks down quickly, those initial messenger RNA molecules go away, and the cell overall stops making insulin because it no longer has the messenger RNA. So people were worried about these new vaccines. They had all these questions about these new vaccines. You guys know how new messenger RNA vaccines are? Do you guys know when the first messenger vaccine was tested in a lab, and it was tested in a non-human mouse model? Any ideas?
20, 30 years ago.
1982. They're new because this is the first time we've been using them large scale in the treatment of viruses and things like this. They've been being used for a number of years in the treatment of cancer, as it turns out, because you can put a messenger RNA molecule in a person that will then cause that person's cells to produce the markers that are on the outside of cancer cells. And those markers then activate the immune system and basically gets your body to attack the cancer cells. So it uses your own immune response to basically get rid of cancer. So they've been using them in cancer treatment for a while, but this is the first time that we've been using them in something like a pandemic. But the cool thing about them is that you're not getting a full viral particle. You're getting just a little snip of messenger RNA, and the only thing that that messenger RNA does is it tells your cells to make this protein that is on the outside of a viral particle. And when that protein is floating around, your immune cells in your blood system come into contact with that protein. They recognize it as a protein that doesn't belong to you, and it mounts an immune response to that protein. And that protein just happens to be the protein that's on the outside of the viral particle. So then when your immune cells come in contact with a viral particle, it attacks it because it's been primed, it's been sensitized to that particular protein. The cool thing about messenger RNA molecules is that when they get into your system, they create some of the protein, but they break down relatively quickly. And now you have those proteins circulating inside of you, but you don't have this big buildup of those proteins because messenger RNA molecules are unstable and they don't stick around for very long. This is one of the reasons why people were worried about were these things actually going to be effective, because you get this messenger RNA in your body for a short period of time. Does it produce enough of the protein to actually cause your body to mount an immune response? And so they had to do studies to see if it was actually effective. So people were really worried about this. They had all these questions, but if they had only sat through the Ecology of Food at William Jewell College, they would have learned about this process by which DNA is used to form messenger RNA is used to build proteins. They would have learned that DNA is really stable because you can activate DNA over and over again to create messenger RNA. Messenger RNA is unstable because if it was as stable as DNA, you would be pumping out a lot of proteins that you don't actually need all the time. And in some cases, those proteins, if they get found in too great of abundance, it can actually have negative effects on you. So messenger RNA is transient, sticks around while it's needed, it breaks down, and then if it's ever needed again, you can go back to the stable DNA and recreate more messenger RNA to create more protein. One of the reasons that you take science, technology, and human experience courses as part of your training when you're not a biology major or a chemistry major or a physics major is because knowing some things about science is actually pretty useful. In the same way that me knowing some things about economics is actually pretty useful. I know way more about economics since I've been teaching this class than I did before I started teaching this class, because I realized that I needed to learn some things about economics. So I went over to the people in the economics department and I said, hey, I'm trying to understand these things, can you help me understand these things? And I'm better off for it. I just bought a car last night. I'm glad that I have some economic knowledge because I was able to go around and search for my own loan. I had loan offers from the people who were selling me the car. I went to my own bank, USAA. I recognize what annual percentage rate means. I know how percentage rates get applied to loans. And I was able to get a much better deal getting a loan from my own bank where I usually bank than from the car dealership, et cetera, et cetera. In a pandemic, this thing on the board is useful to you because you're getting all kinds of reports from the news that if you have a little bit of knowledge, you don't have to worry about what they're saying on the news. You can parse what's being said on the news and say, okay, well, I know that this is how protein synthesis works. So when I hear the word mRNA, I know where that fits in, and I can use my own knowledge that I've gained through my education to say, maybe I don't have to be worried about this thing as much as people are saying. Why am I bringing all this up? Because I'm not trying to be political about this, I'm trying to be sciencey about this. Yesterday on National Public Radio, they had this it was a colossally stupid story. Sorry, I just said NPR is colossally stupid. They were talking about boosters. You've been hearing about boosters? Boosters. Biden and Fauci are saying, everybody should get boosters. If you've already had the vaccine, in case the effectiveness of the vaccine wears off, you get a booster. You get boosters for all of your vaccinations. When you were a little kid, you grow up, and when you're in high school or college or an early adult, you generally go back and get a booster. And what it does, it just gives you the vaccine again and reminds your immune system that, oh yeah, there's this thing that I need to be worried about. When your immune system gets exposed to a foreign substance, like the antigens, these proteins that coat the outside of things like viruses and bacteria and things like this. When your immune system gets activated, there's an initial mounting of an immune response, but then there are these special types of cells in your bloodstream called plasma cells. And sometimes, at least back in the old days, back in the 80s, we referred to these as memory cells. And these are cells that last a lot longer than your normal white blood cells. And what they do is they carry with them that sensitivity to that particular antigen that they came into contact with. And these memory cells are useful for mounting an immune response because they can mount an immune response much more quickly because they've been sensitized to this antigen in the past. And they hang around for a long period of time. They don't live forever. Your red blood cells, you cycle through those really quickly. You're constantly making new red blood cells. Your immune cells in your blood, white blood cells, stay around for a much longer time, and these plasma cells, these memory cells, stay around for a really long time. But they do eventually go away, and so when you get a booster, what you're doing is you're reactivating those plasma cells, but then you're also creating new plasma cells that then remember the exposure to that particular foreign substance. And usually for most vaccines, you get a booster like 10 or 15 years after you got the initial vaccine. It's a long period of time. So Biden and Fauci made these comments this week saying, oh, we should give boosters to everybody to make sure that their immunity stays for the long term. And they had this interview with these 3 science reporters at NPR, and they presented the 3 reporters as 3 reporters taking 3 different approaches to thinking about boosters. One reporter was reporting on a CDC study that basically shows that even after 9 months, the COVID vaccines that are out there are still pretty effective. They were 90+% effective when they were initially given to people, and they're about 80% effective at avoiding getting seriously ill after 9 months. So it's only a drop of about 10%. And in terms of keeping you out of the hospital and out of the ICU, they're essentially as effective as they were before. And then they talked to this other guy who does global health reporting, and he's like, we shouldn't get the booster because places like Africa have 1% vaccination, while we're up at higher than 50% vaccination. We don't need to be wasting doses of the COVID vaccine on people who are already vaccinated and want boosters. We should actually be getting this vaccine to places like Guatemala and sub-Saharan Africa where hardly anybody is vaccinated. And then they had this third reporter who was like, but people want to feel safe, and they'll feel safer if they have the COVID vaccine, if they get the COVID booster. What's the problem with presenting a news story this way?
There's no evidence, or like, there's no research really.
What was the first reporter reporting on?
I was seeing the like study.
It was reporting on a Centers for Disease Control study that showed what?
Like effectiveness.
Yeah, that effectiveness hasn't really dropped very much in the 9 months that we've been having COVID vaccines. So what's the problem with the way they presented this news article? I was terribly disappointed in NPR. I like NPR a lot. I just don't like what they did in this particular instance. By creating a news story in this kind of interview format where you have these three people taking these three different views, what's the problem with presenting information that way? Yeah.
It makes all the information presented seem like it's on an equal footing.
It makes it seem like it's all on an equal footing. So, if there were plenty of vaccines to go around to everybody, there's no harm in giving Americans who are already vaccinated a booster, because there's plenty of vaccine to go around to everybody, and it probably won't help, but it also probably won't hurt. So if getting a vaccine feels better to you because you'll feel safer, then by all means go and take the vaccine. But the fact of the matter is, we don't have an unlimited supply of vaccine. There are some places in the world that have a lot of vaccine. There are other places in the world that don't have the vaccine. And so that first position, which is get the booster if it makes you feel safer, even though it may not do anything to help you, and it probably won't do anything to hurt you, is kind of negated by the fact that globally vaccines are in short supply. And then the other thing is that the first reporter reporting on studies that have been done and completed by the CDC that shows that the vaccine is still effective actually says you don't really need a booster. The first reporter's position is the strongest position because it's based on evidence. This is what scientists do. They look at the evidence. Which means that the other two positions don't have as much standing because one of them is just an emotional response. I'll feel safer if I have a booster. Okay, you'll feel safer, but you won't actually be safer. How do we know that you won't actually be safer? Well, because the CDC has done studies that show that you're not going to really be much safer if you get a booster rather than not getting a booster. The other argument that global supplies of vaccines are low, and we have lots of countries where vaccination rates are really, really terribly low, that has more stature than the emotional argument, because in light of the vaccine shortage globally, it seems dumb for Americans to be getting a booster shot that isn't even going to really give them any added protection from the virus. So when I hear stories like that on the radio, unfortunately on NPR, which I respect a lot, I cringe because it presents each of those reporters as reporting things that are all equally valid. And the weird thing is that there was also this weird gender bias in it. The three reporters were two males and a female, and the female was the one who was covering the CDC stuff, and she kept being talked over by the two guys, which then made it seem like the other two arguments actually had more weight than the actual data from the CDC. So, I say all this to say, I couldn't say this in prior iterations of the ecology of food. This stuff on the board, you as a citizen in the United States should know this. I have never said that in an ecology of food class. You, as a citizen, should know this. It's why I teach it, but it's never been as important now to know these kinds of things. It's never been as important as it is now for people in general to know a little bit of something about basic biology. And in this case, it's kind of complicated basic biology. But one of the things we should be teaching you at Williamsville College is we should be teaching you how to deal with complicated information. If you're having trouble dealing with complicated information, come and see me. None of you have been to see me in my office. My biology majors, I cannot beat them away from my office door with a stick. So, if you're having difficulty with this information, by all means, come and see me. Mutations. Mutations are errors that appear in DNA. They appear in the nitridized base sequences of DNA, and they're random. They just pop up. It's like a copying error. When the scribe is sitting there making copies of a book, he introduced new elements to it. Maybe he didn't like that particular illustration, so he embellished that illustration. But then he also made all kinds of inadvertent errors, just like when you're copying your notes, for example. If you copy notes as a way of studying, errors may get introduced into your notes because you misspelled a word or something like that. So these errors are random, they're undirected, they just happen as you're copying DNA. Remember that you can transcribe DNA into messenger RNA, but you also make copies of your DNA during cell division, so you're also transcribing DNA into DNA when you're getting ready for cell division. So whenever you make a copy of something, you can make copying errors. The cells make copying errors. These copying errors can be substitutions, where you trade out one nucleotide for another nucleotide. You replace an adenine with a cytosine or something like that. So you just, the cell meant to put an adenine in there and put a cytosine in there where an adenine was supposed to be. In some cases, there are deletions, where you were supposed to put a nucleotide in there and it didn't get in there. You just skipped it. In some cases, little duplications occur. You might take, you have a adenine, thymine, guanine, cytosine, cytosine, guanine, cytosine, thymine, and as you copy this into a new strand of DNA, you accidentally get a duplication of the GC, so now the genetic code has a little duplicate in there. Duplications, insertions. In some cases, there are these things. This happens in corn a lot. It was actually discovered in corn by this woman named Barbara McClintock. They have these things called transposable elements. It's where big chunks of your chromosome will break off, go floating around in the cell and become reattached to another chromosome. And they do this kind of by design. They have an evolved tendency to do this. And what it does is it mixes up your genetic material, increases the rate at which kind of mutations occur, which then increases genetic variation, which might have been useful to corn at some point in its past evolutionary history. The rate at which these mutations occur is really low, about 1 in a million base pairs change for each duplication. So if you have 3 billion nucleotides in your DNA, you replicate that once, 3 billion divided by a million, that's, what is 3 billion divided by a million? Yeah. 3 billion, we can do this. Yeah, so if you have 3 billion nucleotides in your body, you duplicate your DNA once, you can expect there to be about 3,000 errors. That's a lot. Well, it's because you have a lot of nucleotides. If your mutation rate is 1 in a million, which is a pretty standard rate that we throw around. But it varies a lot. It varies by taxon, so some organisms have higher mutation rates than other organisms. It also is not evenly distributed in the genome. Some parts of the genome experience mutation rates at higher rates than other parts of the genome. So mutation rate is a tricky thing to deal with. What's important from your perspective is what happens when this happens. So substitutions are sometimes referred to as single nucleotide polymorphisms. That's what a SNP is. It's just trading out one nucleotide for another nucleotide, trading out an adenine for thymine, or whatever. And these things oftentimes don't have any effect on the protein that is formed, especially if that single nucleotide polymorphism occurs in the third portion of the sequence of the codon. Because remember, there's a lot of redundancy in the genetic code, and those kinds of changes in that third position don't necessarily change the amino acid that is specified. Some of these are transitions, where you trade out a pyrimidine for a pyrimidine. Remember that we have two types of nitrogenous bases. One of them are single ring structures, one of them are double ring structures. Pyrimidines, which are cytosine and thymine and also uracil, are single ring structures. And it's easier to trade out a single ring structure for a single ring structure because it doesn't change the diameter of the DNA molecule when you do this trade out. That diameter stays the same. And one of the things that your cell does when it checks its DNA after it's copied its DNA, is one of the things that your cell does is it checks the width. And if it changes width, it actually will go in and fix that mistake. So transitions, which are trading out pyrimidine for a pyrimidine or a purine to a purine. Purines are the double-ringed nitrogenous bases, so they make the molecule wider. You trade out a double ring with a double ring, it stays the same diameter. These are way more common because of that reason than what we call transversions, which are trading out purines for pyrimidines or pyrimidines for purines. Because when you trade out a purine for a pyrimidine, it was a purine and now it's a pyrimidine, the molecule is less wide. You trade out a purine and put it in place of a pyrimidine, it gets bigger. That changes the diameter and the cell can sense that and go in and fix it. Deletions and additions, though, are more catastrophic. And it's because when you take something and put a new nucleotide in there, this codon is the same, but this codon is now different. And that affects every codon from there on down the length of the gene. So these things are called frame shift mutations, because putting a new one in or taking one out, everything shifts down. And so the codon structure, this 3 nitrogenous base structure of messenger RNA, gets altered when you remove or add a mutation to DNA. And I have examples here. So we have DNA, and there's a sequence here. We can figure out what the sequence of messenger RNA. AUG, remember, methionine is the start codon of messenger RNA. And so this thing will code for a very, very short protein with only 4 amino acids, methionine, lysine, phenylalanine, and glycine, before it gets to this stop codon. But if something happens and we put an extra adenine in here, it adds an extra uracil in here, and so it immediately stops protein sequencing because, or protein synthesis, because what used to be a codon that coded for lysine has now been converted into a stop codon because it added this extra A, and then that made this first codon different, and it made it different in a way that stopped protein synthesis all along. The other thing that can happen is if you have a deletion or an insertion, you lose the A here, which means that you lose the U here, And so the first two amino acids are correct, but all of the amino acids after that are not correct for this particular protein. So in some cases, these deletions lead to a loss of function because it just doesn't even make the protein at all. Or if it frame shifts but it keeps doing protein synthesis, it's referred to as a missense mutation because even though it's making a protein, it's a protein that doesn't have a function because the amino acid sequence is incorrect. Because losing that or inserting that nucleotide caused the whole codon structure to either shift up or shift down. So all the amino acids after that are not the correct amino acids. So these types of mutations are much more catastrophic from the standpoint of making a protein
than just a simple nucleotide substitution mutation.
So mutations are different in terms of their type, and the consequence of those differences is that some mutations cells can weather pretty easily, other mutations have more catastrophic effects. Does that make sense? Okay. We did not get to genetics at all, but basically what we've done so far is we've done this. So this is kind of a summary slide. DNA to RNA is transcription. DNA is used as a template to make RNA, but you're still in the language of nucleic acids. Translation is when you take the sequence in messenger RNA. It specifies the order of amino acids in a protein. It's like changing languages from the languages of nucleic acids to the languages of proteins, which are amino acids. But this is basically how we go from the genes that are found in DNA to the phenotype, which is represented by proteins. Proteins are what does all of the work in the bodies of organisms. And so as a result of that, if you change the proteins, you change the ability of the animal, the organism, whether it's a plant or an animal, to do things and do things in the ways that they do them. It changes not only your physical appearance, but it also changes your physiology when you make changes to the proteins. So this is super important because it has to do with what you look like, how fast you run on the track, whether you're a good basketball player, et cetera, et cetera. But all those proteins would not be formed without this process of what's known as the Central Dogma. So we didn't get to any genetics. So when you look at the first exam, all that working out genetic crosses and stuff, that's not on there for Monday. Questions?
Would that be transferred over to the second exam?
It'll be transferred over to the second exam. I don't like moving exam dates because they're on the schedule and you plan for them. Like, oh, I have an exam on Monday in College of Food. So rather than changing the exam so that the exam covers the same thing that last year's exam one did, I just changed the coverage of the exam and left the exam date in place. All right.